So now that we've given a few more minutes to get in and have our participants join us, I'd like to tell you that over the next 30 minutes, we're going to hear from Dr. Natasha Kekry, who for me is in my top left, Dr. Bernard Thébault, who is on my top right, Maria Madana, who is on my bottom left, and myself. I won't be speaking as an expert on anything outside of introducing you to our experts and trying to guide the conversation. So if I can jump right into it, I'd like to introduce you to Dr. Natasha Kekry. Dr. Kekry is a hematologist, and for those of you who don't know, she treats blood cancers, and she helps blood cancer patients and their families through their needs and care. She also happens to be an associate scientist with the Ottawa Hospital and the Ottawa Hospital Research Institute, and an assistant professor at the University of Ottawa. She trained at Harvard and at the Dana-Farber Cancer Institute in Boston. And she is leading our maiden clinical trial of CAR T therapy for cancer, which uses genetically engineered immune cells to help fight the disease. And for those of you who may or may not know, Natasha returned 2 months ago after bringing a lovely little girl into the world. And so we're delighted to have Natasha here. And she signed up for this before she came back, I think. So thank you, Natasha. I'll move on to my next guest, and I'll move over to Maria Madana. Maria is a first-year master's student in cellular and molecular medicine. Maria comes to us from the University of Ottawa, where she took the, and I'll always mix that, the Translational and Molecular Medicine program at the University of Ottawa. And she found Dr. Rudnicki's lab as she searched for her opportunities. Maria has been involved in Dr. Rudnicki's lab for 3 years and is one of our promising next generation of future leaders in science and discovery. And I'll leave with Dr. Bernard Thébault, who is a neonatologist and senior scientist at the Ottawa Hospital and at CHEO, the Children's Hospital of Eastern Ontario. He is a professor of the University of Ottawa and holds a University of Ottawa Partnership Research Chair in Regenerative Medicine. Originally from Munich, studied and trained in France, and for those of you who'd like to know, played a little bit of semi-professional soccer. At the time, he had to make a decision. Was it going to be a professional soccer player or a professional scientist and clinician? We're delighted that he's chosen to pursue the Nobel Prize instead of the Champions League trophy. So welcome, Dr. Thébault. Happy to have you. And more importantly, he leads one of the only labs in the world investigating cell and gene therapy for premature babies, which recently launched its clinical trial. So perhaps if I can, I'll jump right into the questions. It's a question for everyone. Perhaps Dr. Kekry, you'll go first. Maria, you'll go second. Dr. Thébault, you'll go third. If I can lead with the question, could I ask each of you to please discuss your specific work and field of discovery? Dr. Kekry, can I go to you?
Great, thanks, Rob. And thanks everyone for joining us today. So as Rob mentioned, I'm a blood cancer doctor. So I see patients with certain types of cancers, mostly leukemias and lymphomas. And unfortunately, I work mostly in stem cell transplant, which is an incredibly aggressive therapy for these patients. And in many patients, they don't respond to therapy, unfortunately, and have to succumb to their disease. That motivated me to work in this area of cellular therapy called CAR T cells, where we use a patient's immune system, their T cells, and we re-engineer them in the lab to target their cancer cells. And we try to do that in a way that allows them to respond when they've not responded to our standard therapies like chemotherapy, radiation, and transplant. In doing so, we've been able to create a Canadian program for making these CAR T cells. And we are always working on new targets for both patients with blood cancers, but other types of cancers, and finding ways to make this a more innovative and feasible approach for our patients in the Canadian setting. And that's essentially what has been my work since starting at the Ottawa Hospital in working in these CAR T cell therapies. I'll leave it at that for now, Rob.
Thanks, Dr. Kekry. Maria, maybe I can, if I'm deviating from my own pattern, it's just to show you that this is live and not pre-recorded. Maria, can I ask you to go next, please?
Yes, absolutely. So hello, everyone. Thank you for joining us today. As Rob mentioned, I'm Maria. I'm a first-year master's candidate in Dr. Michael Rudnicki's lab. Dr. Michael Rudnicki is the senior scientist and the director of the regenerative medicine program here at the Ottawa Hospital Research Institute. And we currently study skeletal muscle stem cells, the biology of these skeletal muscle stem cells, and a specific degenerative muscle disorder known as Duchenne muscular dystrophy. So DMD is a lethal genetic disorder that affects approximately 1 in 3,500 boys. And due to this severe progressive muscle weakness and degeneration and respiratory issues, patients with DMD often die before the second or even third decade of life. And so our lab has made many important discoveries with respect to DMD. We found that DMD is actually a skeletal muscle stem cell dealing with these skeletal muscle stem cell issues. And we actually found that there's, in patients with DMD, there's this imbalance in divisions of their skeletal muscle stem cells. And on top of this, we actually found a lot of therapeutic candidates that can help to reinstate that balance. And so we found all of these discoveries in mice. And so my project is actually confirming these important discoveries in a human model. And what I mean by this is we actually took cells in a non-invasive way from patients with Duchenne muscular dystrophy, and we've actually genetically reprogrammed them to become stem cells. And we've taken these stem cells and then transformed them into skeletal muscle stem cells. And that has given me a platform to work with and trial these therapeutic candidates that I was talking about earlier. So we're still on the very basic side of research, but with my project, the hope is that we can move into the preclinical stage after confirming these important things.
That's incredible. Thank you so much. Dr. Thébault, can you finish on this question and we'll move to the next one?
Yeah, thank you, Rob. So I take care of critically ill babies in the intensive care unit. And unfortunately, complications of extreme prematurity, babies that are born too early, is now the first leading cause of death in children under the age of 5 years. And so in our lab, we are developing what I believe are game-changing therapies to curb this complication of extreme preterm birth. And we take advantage of cell and gene therapies to do that. And one exciting project that is now moving from the lab into patients is the use of the umbilical cord, from which we extract so-called mesenchymal stromal cells, or MSCs. And in the lab, we have shown that these MSCs can prevent lung injury, one of the most common complications of preterm birth. And we have now launched the first clinical trial in North America, testing these mesenchymal stromal cells for their safety in extreme preterm infants. At the same time, we're working already on the next generation of cell therapy. And we have also very exciting data in the laboratory with a gene therapy to cure a lethal lung disease, which is rare, but for which there's no therapy. And we're currently developing this gene therapy product to go into patients, hopefully within 2 years.
That's incredible. So I am just a regular person with, I went to a small university with not a lot of science background. So I'm going to ask a second follow-up question. And I'm going to ask each of you to break it down for me as though you're speaking to me in the backyard when the world opens up again over an iced tea. We've heard about your science, but now I want to hear what the most exciting element of your specific work is, and why you're what you're most optimistic about the difference it's going to make for patients in the future. And perhaps in this case, I'll switch up the order a bit. We'll go, Maria, even though you mentioned your basic science, what might this finding and discovery mean 20 years, 15 years down the road? What kind of outcomes? And then Dr. Kekri and Dr. Thébault, if it's okay, I'll go there next.
That's a great question. Thank you. So I forgot to mention earlier that Duchenne muscular dystrophy actually has no cure. So it's just a very debilitating disorder, and there are corticosteroid treatments that help slow down the progressive muscle weakness, but in the end, it just slows down the disease. There's no cure. But our lab has found a very specific area of this disorder that can be targeted by a drug that has the potential to be a game changer in therapeutics for DMD. I think, with my project, I'll be able to show people that, hey, yes, it does work in mice models, but it can also, this drug can also work in cells that have come from patients with DMD, that the relevance, I guess, is just greater. And so, yeah, just testing this therapeutic, and it's just a step in the right direction, at least, to helping these patients with this severely debilitating disorder.
I think that's a great description, Maria. I think that sometimes when we get all excited about what's going to hit the clinic tomorrow, we forget that these discoveries were found 15, 20 years ago. And so I think that, okay, so I'm just reading the comment about increasing my volume. So if I can ask the speakers to really project in your privacy of your own bedrooms or offices, wherever you are. So I think that, thank you, Maria, because it's important for us to know that these discoveries were identified years ago and they've started to get into humans. So Dr. Kekri, with that now being a natural transition, can I hand the torch to you?
Sure. So for CAR T cells, which is what I work on, and making these engineered T cells for cancer, patients with leukemia and lymphoma who don't respond to standard therapies, we now have an option that has shown response rates anywhere from 50 to 80%. What I'm working on specifically is how do we build a sustainable model of finding the next generation of CAR T cells so that we can cure more people and make it a safer and more tolerable therapy for patients. So in Canada, we are working with a bread box type of machine where we spike in all the components we need, and then about 10 days later, it puts out these CAR T cells that we then give to patients. We're working on making that system run within our Canadian programs, cancer programs, so that we find a more feasible way for this program to run. Because right now, commercial CAR T cell products, in the US and other places, cost anywhere from $400,000 to $500,000 for the product. That's not including clinical care. And I think in Canada, we have great science that we can find not only new ways, new targets for these CAR T cell therapies for newer other types of cancers and other patients, but also we can build this into a Canadian system where we can make it sustainable for Canadian patients to have access to these therapies. So I think what we call our program is it's CAR T cells with some maple syrup on it, because we are doing it in a way that we think is more sustainable in the Canadian system.
That's incredible. And I'm going to come back to that later, because one of the elements that people won't realize is that we're doing that here in Ottawa. We have the facilities in Ottawa, and we're not dependent on a pharmaceutical company or a government to allow us to discover true findings. So that's incredible. Thank you, Natasha. Dr. Thébault, can I pass it to you?
Yeah, thank you, Rob. This addition was a perfect segue into the discoveries that we're making in our lab. And there's one quote that is attributed to Bob Kennedy that really exemplifies what is so exciting about our work. And the quote says that some men see things as they are and ask why. And we are able to dream about things that never were and say, why not? And then we can make it happen in the lab. And so these cell-based therapies, we can explore them in the lab. And because we have the manufacturing capabilities, we can make them ready for clinical trials. And we also have an amazing method center that designs clinical trials, allows their completion and their analysis. And so we have the full pipeline to go from the discoveries into patients. And the most exciting part is, of course, the one that we're now doing in the clinic, where we will be able to test if these cells have lung protective capabilities. But at the same time, because these cells, unlike pharmacological drugs, these cells may have pleiotropic effects, and they may protect the lungs, but at the same time, the brains of these vulnerable babies, and we may affect overall outcome of these extreme preterm infants. And as a consequence of our research, we were also able to collaborate with Duncan Stewart, who's now leading a clinical trial using these exact same cells from the cord to treat patients with severe COVID-19 acute respiratory distress.
You use the word consequence, but I think benefit. You know, it's one of those things where it's the side benefit of having an infrastructure at the Ottawa Hospital Research Institute, the clinicians and the medical teams, and then the patients willing to participate in research. It's a phenomenal recipe for advancement and discovery. And we are uniquely positioned in Ottawa. So thank you all three for answering that question. I'm going to take a little left turn at Albuquerque, and I'm going to go into the motivation. Maria, I'm going to start with you. You're in the early stages of your investigative journey. You're smart, and I've done my research. You're smart. You were well sought out. You were recruited by other institutes. You came to Ottawa. You stayed in Ottawa. Why did you choose the Ottawa Hospital, the University of Ottawa, and what was it that lured you away to train here?
That is a great question. And I think I'll start from when I started my research career. So I was in third year of my undergraduate degree in TMM, in translational and molecular medicine. And I had the opportunity to do a 6-week rotation program in Dr. Michael Rudnicki's lab. And it was during that 6-week rotation program that I fell in love with Michael's work. I fell in love with the people that were in this lab. And it was in that time that I really, really learned what the Rudnicki lab does. You know, this lab has spearheaded the skeletal muscle stem cell field. It's a worldwide renowned lab, and it's been doing this since the early 2000s. There have been so many major discoveries in this lab since, like, that dates back to finding the marker that specifically identifies skeletal muscle stem cells, you know, little things about how the skeletal muscle stem cells divide, and of course the discoveries about Duchenne muscular dystrophy and the potential therapeutic candidates. And as a young researcher, I knew that I needed to be in this lab because this is where I will foster my scientific spirit and be surrounded by mentors that will guide me and direct me. And another really important thing that I learned is, as a young researcher, it's very, very important to be able to try different methodologies and see what you're interested in, see what you're passionate about. And at the Ottawa Hospital Research Institute, we have so many state-of-the-art, cutting-edge facilities that are available to us. For example, for my specific lab, like for my lab work, I mainly use the stem cell facility in Dr. Bill Stanford's lab. So this is where I was trained. It's where I grow my stem cells. It's where I take care of them. They're babies. I have to feed them every day. And that's where I turn them into skeletal muscle. And I trial my different therapeutic candidates. And I was able to use these amazing facilities at such a young age. Like, I was able to do this in third year of my undergrad. And I can recognize that not everyone has this opportunity. And I was able to learn these cutting edge techniques and methodologies at such a young age, at the very start of my scientific career. So all of this considered, it's just absolutely mind blowing. It's why I decided to continue pursuing my research career here at the OHRI.
Awesome. That's great. It's fun to see the excitement and the reason for all of our scientists and all of our individuals that have chosen to come here in their mid or late stages of their accomplishments, or in their early stages as they start to plot out their journey. So thank you so much for sharing that with us. And thank you to the donors on the call today. Much of what you funded in the past has been either the key components of these core facilities or these research institutes, or the research and the researchers that you're hearing from today. So I would be remiss if I didn't take one moment to say thank you so much, everyone on the call, for giving and supporting the Ottawa Hospital and its research endeavors. Dr. Kekri, I'm going to pivot and come to you next. You've been a part of some significant advancements already in cancer research. Even before CAR-T came up, you were already pushing some boundaries on some really unique and individual care and research initiatives. But if I can stay on CAR-T a bit longer, it's kind of the next big thing. I'm reading about it in the States. I'm reading about it in Canada. The Citizen did a full profile that we did not pay for. It was all done by investigative journalism. This seems to be where we're going. Can you tell us a little bit more about the unique opportunity that this intervention provides? And what can I or my relatives who might benefit from this treatment expect in terms of a care intervention?
So, yeah, I think CAR T cells are, at least for cancer, going to be part of this immunotherapy pillar of cancer, as we call it. So the ability for us to re-engineer a patient's own cells to target their cancer, and that's what we work on. I think what patients can expect in Canada is that thanks to places like the OHRI, where we have unique expertise, it allows us to make our own CAR T cells in Canada, which is not true around the world. For the most part, these are done in these are made in highly specialized billion-dollar facilities, mostly in the US and a few in Europe. But that doesn't really provide us with Canadian access to innovative care. So, the next best thing, the next best CAR T cell. And I think that what patients can expect is that as we build a network in Canada of great scientists and great thinkers who want to work on this for Canadian patients, I think patients will see more and more of these trials that are quote unquote made in Canada, because we have the facilities specifically in Ottawa to help us in manufacturing of these cells for Ottawa patients, but ultimately also Canadian patients. But that specifically came from expertise and infrastructure that Ottawa has. And I think patients can just expect us to keep building more trials and keep working harder to bring patients access to newer therapies from these CAR T cells that we've seen great success with.
That's amazing. Thank you. Everyone, I'm going to do a little bit of a process check-in because we're about halfway through this session's time. And I want to remind everyone that on the main page of this portal, there was a resources tab. If there's information on this, the science and the discoveries that we've been talking about, when you go back to the event page, there's a resources tab on the left-hand side. If you click that, you can read the bios of our speakers. You can read the research profiles. There's live links to various internet access, Ottawa Hospital resources. I just want to come back to that to say if there's something you've heard or didn't hear, that on the main page that you all started the live stream for, there was a column on the left that said resources. So please feel free to use those as our participants are still with us. That just my little infomercial for 5 seconds. Dr. Thébault, if our guests haven't noticed, there's a little pattern to my asking. The next question's for you. You too have both a balance of care and research responsibilities. You touched on your trials and what you're doing. Can I ask you to expand a little bit more on what trial you're running currently and why is it important to lung development, lung progression, and healthy quality of life development for children as they're babies?
Yeah, thank you, Rob. So our trial is called the HULK trial, Helping Underdeveloped Lungs with Cells. And we chose to target lung diseases because it's the main complication in extreme preterm babies. And this chronic lung disease of prematurity has been described over 50 years ago. And currently we still have no treatment for that. So here we truly have the possibility of finally making a substantial improvement against chronic lung disease of prematurity. And these mesenchymal stromal cells were very promising in the lab. So now it's time to test their feasibility and safety in preterm infants. So it's always exciting. I think the most exciting part of a clinician scientist when you're able to translate the discoveries from the lab into patients. And so this trial will hopefully not only affect the lungs of these babies, but as I mentioned before, because of these novel properties compared to regular pharmacological drugs, because of the properties of these cells, we may also affect other organs in those babies, and most importantly, the brains. So what we expect from this trial is to show overall improvement in outcome of these babies improved lung function and improved neurodevelopmental outcome as well. Now, this is only a phase 1 trial where we are testing safety and feasibility, but because of the foresight of our method center, we included an observational cohort, which can in some instances help us to compare if our strategies make a difference. The sample size is still small, but there might be some indication. And this trial will then hopefully lead to a national Canadian multi-center trial to test the efficacy of these cells. And again, it's the first in North America, and we hope that we will have many more innovations that will have been initiated in Ottawa.
Thank you, Dr. Thébault. Now, for the audience, we have had a couple of questions been asked, and in some cases, for privacy reasons, I'm going to ask a question similar to the one you're asking versus the exact question. And that's just if you have additional care questions. So some of the questions are related to care. So this question is for Dr. Kekri. Dr. Kekri, I am editing the question, and I would ask the person who asked it to look me up in the system, contact me tomorrow, and I will put you in touch with the right triaging environment to ask as a specific question, but I will ask the generic one. Dr. Kekri, how are people referred to the ALL clinic at the hospital, and how might they have their family physician or their oncologist refer to the CAR-T program for future participation in the research?
So I'm fortunate to work with in a small community of physicians. So in terms of hematologists who treat leukemia and lymphoma, we essentially know each other pretty well across the country, especially in Ontario. And so for right now, our trials are open in Vancouver and Ottawa, the Ottawa Hospital, and patients are referred in by their treating physicians. So if a treating physician thinks that a patient with either acute lymphoblastic leukemia or certain types of lymphoma might be considered for our trial, that physician usually reaches out to me or one of my co-investigators directly, and we usually then accordingly will do a consult and decide if that's a potential patient for the trial, if possible. We get referrals from all over, but I would say the majority of them right now are from Ontario and British Columbia, because those are the provinces where we're open. And we are working to expand our network of treating sites for the trial throughout Canada, but that has just started. But really for right now, in Ontario, patients or physicians are well aware of what I'm working on with all of the collaborators we have, and they usually are very versed in how to reach out to us, and I'm pretty accessible and respond to them quite quickly in terms of questions that they usually have. So that's been great. It's been great to have support from my colleagues across the country.
Thank you, Dr. Kekri. And I hope for the individual that asked that question that gave enough guidance for them to recommend to the people they're asking on behalf of to move forward. We'll leave it at that. If not, contact me and I will help triage the answer more formally. The next question, being conscious of time, we have about 4 minutes left before we're being asked to go to the next session. Bernard, I'm going to ask a question to you and to Maria, depending on who wants to answer. There's a lot of hope related to stem cells and stem cell clinics and the concept of what is a valid stem cell treatment and what is something you read about online that you just know doesn't smell right. Can you maybe give us some content about how regular people can tell the good from bad in stem cell interventions? I'll go to Dr. Maria first, and then we'll go to Dr. Thébault.
Okay, I promise my answer isn't that long. I have two things that I would like to point out. The first one is make sure to look at the source. Is the source a reputable source? Is this something that, is an established institution where, lots of good research has come out of? And the second thing is to just be really wary about what they're trying to say. I've seen some advertisements that say this treatment is with 100% efficacy. Yeah, no, biology is usually never 100%. Like that's something that I've learned in my research, that it's never usually like a straight cut answer like that. So those are the two things that I would consider.
Yeah, thank you, Maria. Absolutely. As an individual, one should look at where these cells are coming from. And currently, cell-based therapies are still experimental. So they should only be done, these studies or administration of cells should only be done within a registered clinical trial. And most of the time, these clinical trials come out of university hospitals. And so I would definitely talk to my GP or a physician who works at a university center to provide transparent information about a given cell therapy. And it's important to be informed.
Lovely, thank you. Everyone, we have 1 minute left, and I've got a couple of questions that came in under the wire. So here's what I'm going to say. The first question is, commercial rights, proprietary rights to research that has been identified by scientists at the hospital. There's the question is, who owns the rights? And I think that, Wally, to be fair, the question is, there's a combination depending on who started the discovery, who's continuing it, and who you have to engage to help you expand it. So there's no easy answer to your question. I apologize. It really is a case-by-case situation. And the second question that is from Chandra about algorithm use in late-stage cancer for stem cell treatments. Like, how are you establishing or identifying them? Natasha, is there any way to identify why, or Bernard or Maria, why certain late-stage cancers are chosen for stem cell intervention or biotherapeutics?
Dr. Thibault can probably give a different answer than me, but in terms of cancer, cellular therapies such as CAR T are incredibly complex and incredibly risky, I would say. And I think that we often start, not that I think this is the correct answer, but we often start with patients who have exhausted other options, because we want to first show that they're safe. And oftentimes, to do that, if we do that earlier in lines of therapy, we're competing with patients getting standard of care options where they have a response already. And it's a bit difficult to enroll those patients, even though I think there's a very good chance they would benefit from these therapies. And so that's sort of how this field has evolved. I agree, we started with later stage disease. We probably shouldn't, but that's just the reality of the amount of work that it takes. And you heard Dr. Thibault mention that his first study has to be about safety and feasibility. And that's just the reality of the way research works.
I apologize, everybody. We are going to get the boot, I'm sure of it. So Dr. Thibault, I promise you an avenue next time. Thank you, everyone, for joining us. I'm looking at the clock and wondering why we haven't been kicked out yet. Jay, if you're still on, are we getting the boot? Maybe we got it already. Okay, Jay, thank you so much. Thank you, everyone, for joining. I hope this was informative and look forward to the next time.